Hi everyone, I wanted to pick up with the rest of the Microbiology Chapter 1 PowerPoint. We ended up not being able to finish these last 12 or so slides, so I just wanted to do like 15-20 minutes just talking about them in preparation to help you with future quizzes and HESI. So anyways, we're going to start on slide 34. We're going to talk about taxonomy. So this is literally just a formal system of organizing, classifying, and naming living organisms. So some examples of the way that they're classified is into groups. So domain, kingdom, phylum, class, order, family, and genus, species, and strain. So those are some common terminology that you may see when we talk about taxonomy. There is a scientific name that's given to microorganisms, and this was first established by a scientist named Carolinus Linnaeus, and he actually came up with this binomial nomenclature system, and it was the genus and the species. So the genus is typically capitalized, and it's the first word. So in this case, it would be Escherichia. And then coli is the species, and it is not capitalized. Once you've used Escherichia coli as an example or in a sentence, then it can be abbreviated as E. coli in the future. Here's just an example of the kingdom of life in relation to the bacteria Shigella soni. This is in your book on page 10. If you like visuals, that may be helpful for you to take a look at. Okay, so going back to taxonomy, I talked about how each organism has two names, the genus and the species. The genus is capitalized and the species is in lowercase, and after the first time you use it, the scientific name can be abbreviated with the first letter of the genus. So again, instead of Escherichia coli, it's E. coli. Okay, another group of scientists in 1990 came up with the Woese-Fox system, and this identified microorganisms into three different domains. These three domains that he categorized things into were bacteria, archaea, and eukarya, and then within eukarya exist protists, fungi, plants, and animals. And in his system, instead of using morphological differences to identify, he actually looked at the genetic components of each of the things in these three domains to separate them appropriately. And then in taxonomy, we also want to talk a little bit about Berge's Manual of Systematic Bacteriology. This is a very good resource for bacteria. If you want more information about Berge's Manual of Systematic Bacteriology, you can actually look at Appendix A in your microbiology book, which starts on page 475. That gives you an example of some of the material that's included in Berge's Manual. So you guys, we've touched base on a lot of information just in this first chapter of our microbiology book. You're probably wondering exactly how microorganisms fit into health and disease, although I'm assuming since most of you are LPNs, you already know. So to be honest, we use a lot of microbiology in our daily practice as nurses, and we're going to talk a little bit about that in these remaining 4 or 5 slides, okay? So microorganisms in health and disease. So we talk about microbial ecology, okay? And this is simply the study of microbes and their environment and what that relationship is. So microbes are everywhere in our environment, and they generally have an impact in maintaining ecosystems. So we've talked a little bit about mutualism, commensalism, synergism, and parasitism a little bit several slides ago, but I do want to go ahead and reiterate what the definitions are of these things, because they will be important, and they are important, just basic microbiology terms. So remember, in mutualism, both organisms benefit. In commensalism, the waste product of a microbe provides useful nutrients for another organism. I'm actually going to give you another example of commensalism. So commensalism actually can occur when, for example, when the bacterium Staphylococcus epidermidis uses the dead cells of the human skin as nutrients. With synergism, remember, two organisms are dependent on each other to break down a single nutrient that neither one breaks down alone. So a real-life example of synergism would be when physicians treat bacterial heart infections with ampicillin and gentamicin, two antibiotics, and when cancer patients receive both radiation and chemotherapy or more than one chemotherapy drug at a time. And last but not least, I want to touch base on parasitism. And remember, this is when one organism benefits and the other is harmed. So a good example of this is malaria, which is caused by a protozoan of the genus Plasmodium, is transmitted to humans by the bite of a mosquito. Okay, so now I want to touch base a little bit about normal flora versus pathogens. This is another very important concept in microbiology that I want to make sure that you guys understand. So when we talk about normal flora, what I'm referring to is the microorganisms that live on other living organisms, whether they're human organisms or animal organisms, or just non-living objects, without causing any disease. So keep in mind, the human body is not sterile. From the moment we're born, we become full of microorganisms. We are covered with them. They're in our intestines. And there's about 100 trillion bacteria that form the normal flora of our body. So these are microorganisms, microbes, that live in or on our body that typically don't cause any disease, no illness, nothing, okay? Now, the normal flora is actually helpful to us because it can help us or prevent us from becoming colonized with more dangerous bacteria that lead to infection. So I want to go over actually two examples of where normal flora can turn into kind of a pathogenic situation, okay? And both of these examples being LPNs, I'm sure you guys already know, but the first one is a vaginal yeast infection. So Candida albicans, which is part of our body's normal flora, it lives in our mouth, throat, gut, and it can also live on our skin, is very much present as normal flora of and on our body. So let's say my patient is actually taking an antibiotic for a urinary tract infection. So what may happen is they're given a, let's say, broad spectrum antibiotic for their UTI. However, it doesn't just kill the bacteria that we think is causing the UTI. It also kills healthy bacteria that's in our vagina, which leads to an overgrowth of this yeast. So now we've taken a normal flora situation and we've turned it kind of pathogenic. Another prime example of this is in the case of Clostridium difficile or C. diff. So in several patients, but not all, C. diff or Clostridium difficile is a very normal flora to have present. Typically in patients that have Clostridium difficile in their GI tracts have no issues whatsoever with the bacteria. However, again, if you have a patient who we're using antibiotics on to treat whatever infection that they have, the antibiotics may alter the typical intestinal tract of these patients. And so what can happen is then these microorganisms, Clostridium difficile or C. diff, can actually multiply and literally take over the intestinal tract, causing a pathogenic situation. The thing about Clostridium difficile, and we're going to talk about this in later chapters and in lab, is it's a spore bacteria, and bacteria that are spore-like are very, very hard to kill and very, very hard to treat, which is why you see a lot of patients with C. diff continue to have recurrent infections of it. Okay, we also talk about foodborne disease, and this actually results from consuming food that is contaminated with different pathogenic species of bacteria, viruses, parasites, or microbial toxins. Waterborne. Okay, in terms of food production, many non-pathogenic organisms occur naturally in food, so they don't cause any issues. Wine is produced by yeast fermentation of carbohydrates in freshly harvested grapes, peaches, berries, pears, and other fruits or plants. And then in the production of beer, for example, barley or another grain is used as the source of fermentable carbohydrate. As far as drinking water, the microbial content of drinking water should be constantly monitored to make sure the water is free from any infectious agents. And then as far as the pharmaceutical agents in microbiology, remember Alexander Fleming actually discovered the penicillin-producing mold Penicillium in the 1920s, and he started the successful search for other antibiotic-producing microorganisms. Okay, and we talked a lot about antibiosis, and they talk about that in your book as well. Oops, I think I skipped a slide, guys. Let me see here. Well, I guess I can't go back. Anyways, agricultural microbiology focuses on the relationships between microbes and domesticated plants and animals. Bioremediation is the process of using microorganisms to clean up toxic, hazardous, or unmanageable compounds by degrading them into harmless compounds. Some microbes perform these tasks, and they've been genetically engineered to clean up specific waste or pollutants. And then by a process referred to as bioconversion, microorganisms can convert biomass such as organic matter and human, agricultural, and industrial waste into alternative fuels, including ethanol, methane, and hydrogen. And what I think is really cool is a kind of a new field of microbiology called microbial forensics. And this is actually applied to solving bioterrorism cases, medical negligence cases, and outbreaks of foodborne disease. Your book goes on to further give a few examples, which I actually think are really good real-life examples. They talk about microbial forensics being used in the anthrax attacks of 2001, and then they also talk about microbial forensics being used when you have patients who are purposely injected with HIV or AIDS, and these scientists try to track these microbes to the source. So that was 13 minutes just to conclude the rest of the slides for chapter 1. If you guys have any questions, please let me know. Reach out. Remember, my office hours are Friday from 10:30 to noon, but if you can't meet then, I am more than happy to meet with you at a different time. Thank you.